Q&A box as opposed to the chat box so that we have them consolidated in one place. And now I'm going to pass things off to Michael Richter. Thank you.
Thanks, Laura. So it's my honor to present our grand round speaker for today, Dr. Bryan Kestenbaum, who's coming to us from our own division of nephrology, where he's been a professor of medicine for about 5 years now. Dr. Kestenbaum's been with the University of Washington since he came here as a fellow just over 20 years ago. And in that time, he's been extremely productive as a researcher in the field of chronic kidney diseases, with studies ranging from metabolic biomarkers in CKD to clinical trials of vitamin D and many epidemiology studies. But despite the research contributions, what struck me most reading through his CV was the degree to which he's helped build the careers of trainees. So of the formal trainees he's worked with in the nephrology division and the School of Epidemiology, there are now 7 PhDs, 9 master's degrees, 51 mentored publications, 4 NIH Career Development Awards, and perhaps most impressively, 8 of those trainees have now gone on to be professors themselves. He also serves as the Director of Graduate Education at the Kidney Research Institute and is course chair of epidemiology and biostatistics at the UW Medical School. So in a system where it sometimes seems, to me at least, as if we need to choose between research and teaching, Dr. Kestenbaum's career shows quite reassuringly that this is in fact a false dichotomy. So on behalf of the Division of Rheumatology, I thank you for being here, and we all look forward to your talk.
Thanks so much, Michael. I didn't realize all those numbers, so that was nice to hear. Thanks for everyone for coming to listen to this. I can't see anybody, so I can't notice who recognize people I may know and have come across on the wards and things like that. But thank you all for coming. I think Dr. Elkon sent me an email earlier asking me if I could also make a quick announcement about a course that we're putting on about epidemiology and biostatistics mostly for research fellows, but also potentially for junior faculty to do some basic work in clinical research methods. And that's going to take place starting at the end of February. I think Conrad Lyle sent the email out with the schedule recently, so I just wanted to put a plug in there and have you look out for that and can get in touch with me about details. In a completely different direction here, I'm going to talk about something completely non-epidemiological, more physiological, a kidney physiology talk, really about work we've been doing lately on kidney tubular solute clearance. All right, let's see if I can get this to, there we go. All right, so as we all know, the kidneys do many different things. They perform multiple functions, excuse me, including filtration, secretion, synthesis. But in practice, when we monitor and stage kidney disease, it's done purely based on measures of glomerular function, namely measures of filtration and measures of glomerular integrity, GFR and albuminuria. And these are used to define the presence and the severity of chronic kidney disease, to dose kidney medications, to decide whether to start dialysis, and all kinds of other things. And why do we ignore all the other kidney functions and just focus on the glomerular functions as a means of assessment? A lot of work has gone on behind the scenes over decades to refine these measurements to get them to be reasonably accurate. And they're relatively now they're relatively easy to obtain. Should we be assessing kidney function exclusively by people's glomerular functions? If kidney disease has caused all the kidney functions to decline in parallel, then it's perfectly fine to do so. This would be a theoretical figure of a situation where the glomerular filtration rate is declining at the same exact speed as the tubular secretory clearance and amino acid synthesis and hormonal synthesis and acid-base balance. These are all functions of the kidney tubules. If all these things were to decline in parallel, then one measure would be fine for all. We have reason to suspect that's not the case, that there's individual differences in these functions across diseases and people. Some of that's anecdotal. We know, for example, that when we do kidney biopsies, the degree of pathologic damage often doesn't correspond very well with the level of GFR. Here's two people with the same kidney disease, diabetes, same estimated GFR. The first person has pretty substantial interstitial fibrosis and tubular atrophy on their biopsy. Second person's tubules look pretty good. Now, maybe some of that's due to sampling variability on the biopsy. Maybe some of it's due to the imprecision of estimating GFR, but more than likely the functions are different to some extent among people. Same thing is true about metabolic complications, things like hyperparathyroidism and anemia, which develop as a result of really tubular dysfunctions. And again, these can be highly variable for the same level of GFR. When we do dialysis, really all we're doing is recapitulating a glomerulus. It's just a big filter. It doesn't do all the other things that the kidneys do, like maintain acid-base homeostasis or secrete or do remote signaling. And there's really no question about clinically which one does better. Everybody wants a kidney transplant and the wait list just keeps growing. So certainly there's something important about the other non-glomerular functions of the kidney that are relevant to health. So roughly 5-6 years ago, I became interested in thinking about the problem of trying to estimate other kidney functions, at least for the purposes of in clinical practice, to get a more comprehensive assessment of kidney functions, and to see how those things might apply across diseases in people. And the area I was particularly interested in was the secretion, the tubular secretion of protein-bound solutes and drugs. So a quick review of renal physiology here. All this cardiac output goes to the kidneys, and normally about 700 mils per minute of plasma is delivered to our 1 million glomeruli per kidney. And as the renal plasma circulates through the glomerular capillary bed, about 20% of that is filtered. And the remaining 80% of the blood flow to the kidney is then shunted to the post-glomerular circulation. And it's there that the tubules secrete actively substances from the blood into the urine. So there's sort of two ways for toxins to get removed from the body. One is for them to be filtered and then enter the kidney that way, or they could be actively secreted by the tubules from the blood side into the proximal tubules and then into the urine. The secretion of solutes is quite complicated. So whereas glomerular filtration is sort of passive, it's a filter, it's got a glomerular filtration barrier, certain size things can fit through, certain things can't. Secretion is complex. Oftentimes the secreted solutes are protein bound, so they have to come off of albumin, diffuse into the interstitial space, and interact with the correct transporter on the basolateral surface of the proximal tubule cell. There's a bunch of different secretory transporters that are present there. And then there's an active coordinating process by which these solutes are transported across the proximal tubule and then actively secreted with energy into the urinary space. There are some broad differences between filtration and secretion in terms of what is being removed from the body and how rapidly that's occurring. As we said before, because filtration only filters about 20% of the blood delivered to the kidneys, it takes a lot to remove something that's highly toxic. It would have to sort of go around the body 5 different times to fully get rid of it. Secretion is much more efficient. The transporters on the basolateral surface can remove almost all of a substance from the blood in a single pass through the kidney. So there's much more efficiency in solute removal, potentially being important for removing the most toxic substances. Filtration is unable to remove things that are bound to albumin due to the size limitations of the glomerular filtration barrier, but secretion is, through dissociation of albumin and into the interaction with the transporters, it is able to remove protein-bound toxins and solutes. And because most medications are at least partially protein-bound, if not highly protein-bound, secretion is actually the primary kidney mechanism of renal drug elimination. So how do we go about trying to measure tubular secretory clearance? In theory, it's easy. We find an ideal secretory solute, one that's potentially not filtered to any appreciable degree, is avidly secreted by the tubules, and can be measured easily. And so we would measure the kidney clearance of that substance and that would be a marker of secretory clearance. So we set out to try to find such solutes. We were interested in trying to first come up with tests based on endogenous solutes, because providing or injecting exogenous things like iohexol is used for GFR tends to be impractical. So we're trying to find the next creatinine for secretion, basically something that we could measure that was produced in the body. And we used a whole bunch of different lines of evidence to try to find such solutes. One line of evidence is to find solutes from the literature that were avid substrates of known basolateral transporters on the proximal tubules. And there are many such substances that are strong substrates of these transporters. Here's two examples, indoxyl sulfate and hippurate. We also looked at animal models in which the transporters were knocked out, and they did blood profiling to see which compounds in the blood increased the most, as a clue as to which compounds were most heavily influenced by secretory clearance. We also looked at studies like this. This is a really clever study done by Tammy Search's group at Stanford, where they just asked 7 healthy people to come in and provide a 24-hour urine sample. And they did mass spec profiling on the plasma and urine to calculate the clearances of something like 1,200 different endogenous substances. Not surprisingly, they found the average creatinine clearance in the healthy volunteers was about 140 mils per minute, similar to that of what's expected for a filtered substance or GFR. But they found many substances in which the kidney clearance was much, much higher than that. Again, consistent with secretory clearance, tubular secretory clearance, which can almost completely clear the blood of a substance in a single pass. You can see compounds like titroglycine, hippurate have very, very high kidney clearances, strongly suggesting that secretion is the primary modality of renal elimination. There are also some studies out there, not many, in which direct sampling from the renal artery and renal vein is performed. And we were looking in studies like this for compounds in which there's a large drop in the concentration of the substance from the artery to the vein, again, suggestive of secretory clearance as a primary modality of renal elimination. And this gave us some clues as to some substances like hippuric acid, again, kynuric acid, that were all removed from the kidneys in a single pass to a much higher degree than creatinine. Based on these studies, we came up with a set of, we thought of as good candidate secretory solutes. We called it Secretion 1.0. We did some studies in our local cohorts using that set of solutes. And then over time, working with our laboratory medicine colleagues, and doing more review of the literature, we expanded our panel to this group of candidate markers of tubular secretory clearance. It would be impossible to go on without acknowledging the incredible work of our collaborators, Andy Hoofnagel in Department of Laboratory Medicine, who devised the mass spectroscopy assays for these solutes. And he was able to develop internal standards for all the different compounds that we were, or to buy internal standards and make labels for all the compounds that we were investigating. And also able to determine actual concentrations of these compounds going to upper campus using nuclear magnetic resonance, so that when we measure things like hippuric acid or kynuric acid or xanthosine, things that are candidate solutes, we can get back an actual mass in milligrams per deciliter or nanograms, as opposed to getting back some arbitrary units. And this requires getting a definitive measurement of the amount of substance in the chemical. This sort of summarizes our laboratory data. We were unable to detect 3 of our candidate solutes just because they were below the limit of detection in the lab. And this was sort of we're trying to devise assays that were a good compromise to get as many of the compounds as we could. And so these 3 were lost in the process. But otherwise, for the other compounds, the laboratory characteristics are pretty good. Most of the inter and intra-assay coefficients of variation are relatively low. To further explore the laboratory properties of these compounds, we did studies in people who contributed blood samples over a short and long period of time to understand biological variability in the compounds above that of of the variability seen in the laboratory. One such study was this one we did in 26 healthy people that were in a trial of omega-3 fatty acids. And these participants had no history of any kidney disease or diabetes or cardiovascular disease. And we obtained samples from the baseline examination before they got one of their dietary treatments. We chose this study because at each visit, including the baseline visit, the participants were kept in a hospitalized setting, actually, and an indwelling catheter was placed and blood was drawn every 30 to 60 minutes over 24 hours. So this was an ideal place to explore biological or diurnal variability in the solutes of interest. So one thing we first looked at was just creatinine. We were just interested in, you don't really usually get creatinines every 30 minutes in people. Just how much biologic variability is there in creatinine? The answer is very, very little. Coefficient of variation, biological coefficient of variation over 24 hours, 7%, which is really nice. They didn't know all these nice things about creatinine when they found it 100 years ago or 80 years ago. But it's one of its nice properties. It's a very large amount of it in blood, and it doesn't vary very much, even with eating. Such was not the case with some of our secretory solutes. This is one example of hippurate. This varies all over the place, does increase after lunch, and the coefficient of variation was 52%. And so this is going to make hippurate a non-ideal endogenous solute for measurement of secretory clearance, which is okay. We were expecting, to start with a big list and come down to a smaller group. This shows the biological variation in the solutes from these 24-hour studies, and some of them are okay. Nothing's as good as creatinine. They all have some degree of diurnal variation. They circulate in relatively low concentration, so this is going to be an inherent limitation, at least of the first set of solutes that we selected for study. And just to clarify, when we use these solutes in studies to estimate tubular secretory clearance, what we're going to do is we're going to calculate the kidney clearance of that solute using the equation urine concentration times urine volume over plasma concentration, the UV over P equation, which calculates the amount of kidney clearance in mils per minute. And this is in a calculation that should be theoretically independent of the plasma concentration, if that plasma concentration is relatively stable over the urine collection time. And we've seen for some of our solutes that may not be the case. And this summarizes the kidney clearance in normal people of our candidate solutes. We, from the list of I showed you before, the secretion 2.0, we dropped off 8 of those solutes, either because we couldn't measure them well in the lab or because they had too much diurnal variation. This is sort of the candidate list we went into our studies with, expecting that we would probably drop some more out over time. And you can see that the clearance of most of these is very high, much higher than that of creatinine. For some of them, the kidney clearance is actually quite low, like indoxyl sulfate and p-cresol sulfate. But those compounds are over 95% bound to albumin in serum. So any kidney clearance of those compounds is almost certainly due to proximal tubular secretion, as they can't cross the glomerular filtration barrier. And here's the protein binding data. And you can see for chimeric acid, p-cresol sulfate, indoxyl sulfate, and cinnamyl glycine, the protein binding is quite high. For other compounds, the protein binding was low. We thought all these compounds were highly protein bound when we selected them, but it turns out when we did our own experiments, we found things that were different. Still, in combination, this all sort of works. The highly protein bound compounds are the ones that tend to have sort of lower kidney clearances, where we can still be certain or reasonably certain that clearance is due to tubular secretion. Whereas these other compounds like isovalerial glycine that has very little protein binding, that's the compound where the renal clearance was over 500 mils per minute. So even though there's some filtration of isovalerial glycine, the majority, because of the secretion being, the total clearance being so high, the majority of that elimination is most likely due to tubular secretion. Okay, so those are our candidate solutes. Okay, so that's kind of a long background, but I wanted to sort of introduce where we were and now talk about a couple of studies that we've done with our estimates of secretory clearance. And one of the studies that we've done that I really like, we've got a lot of studies to talk about that we've done with secretory clearance. One of the ones I like is this one called PROCLAIM, because we did it here, and we're able to address a question I've been interested in for a long time, which is how do the kidneys actually eliminate drugs, and how should we be measuring that? So for the most part, secretion is the primary mechanism by which the kidneys eliminate drugs and metabolites. Here's just a partial example of the many drugs that are substrates for transporters in the proximal tubule. Despite that, we dose drugs based on either GFR or an estimate of GFR, like creatinine clearance. And so we're assuming that in people that their level of GFR, or level of glomerular filtration, is tightly linked with that of tubular secretion, so that we can estimate the drug clearance using a different measure of kidney function that we hope is marking the measure of kidney function we're really interested in. So we wanted to challenge that assumption with this study. It's really never been looked at empirically. So the goals of the study were to perform empiric pharmacokinetic profiling of avidly secreted medications across a wide range of kidney function, and to compare our direct measures or our direct estimates of tubular secretory clearance with GFR for predicting the renal drug elimination. So for this study, we recruited 54 people, including healthy people and those with chronic kidney disease, across a wide range of GFR. We excluded people who were on dialysis or had a transplant or had nephrotic syndrome. And in the first phase, we gave them two prototype secretory drugs. One was furosemide, which is 95% bound to albumin. Very little, if any, crosses the glomerular filtration barrier. And it's avidly secreted by the organic anion transporters into the tubules. The second drug we administered was PENCICLOVIR. This one is less protein bound. It's a substrate of a different organic anion transporter and is also avidly secreted with a renal clearance that exceeds GFR by a considerable margin. We measured the kidney clearances of these drugs, as well as our secretory solutes, over a 10-hour timed study visit that included a timed urine collection over the same period of time. And we performed direct measurements of GFR using iohexol disappearance. So we injected people with iohexol and watched the levels decline over time in the blood and used that to calculate GFR. And we're getting experience over time working with our different secretory solutes and have come up with a way of defining a summary secretory clearance score as a scaled average of the kidney clearances of these secreted solutes. So I can show you the individual data as well, but this is just a way of summarizing all the secretory clearance into a single score that we think probably best estimates secretory clearance. So here's the characteristics of our participants. They were on average 55 years old. Their average GFR was 73 mils per minute. We had some people with low GFR, medium, high, and higher. And that was by design. We didn't have enough people in a study like this to try to sample by the type of kidney disease, so all comers were included. 11% had diabetes. There was very little albuminuria. And the calculated clearances of these two medications that we administered, furosemide and PENCICLOVIR, were 120 mils per minute for furosemide. So just, again, this is a renal clearance that's higher than directly measured GFR by about 50%, and yet the compound is entirely bound to albumin. So all of this is tubular secretion, or at least the vast, vast majority. PENCICLOVIR only 20% protein bound, so some is filtered, but again, the renal clearance is much, much higher than that of GFR, again suggesting that the majority of its renal elimination is secretion. So, excuse me, sorry. One of the, I guess I'll start off. This is one of those stages where we found some exciting things and then some puzzling things. But that's great. That's research, and it can't always be all good. Here's the good news. So we spent all this time developing these assays and making this summary secretion score, and we go into the study and we directly measure now renal pharmacokinetics, and we compare our secretion score, which we think estimates secretory clearance, with the direct renal elimination of avidly secreted drugs. We find a pretty tight linkage. For furosemide, the correlation between the summary secretion score based on our endogenous solutes and the actual kidney elimination of furosemide was 0.84. And for PENCICLOVIR, that correlation was 0.88. So our measures are capturing secretory clearance, at least as indicated by the elimination of two avidly secreted drugs. However, turns out that GFR, this is directly measured GFR, also correlates reasonably strongly with the elimination of these two secreted medications. For furosemide, the correlation was 0.77, and for PENCICLOVIR was 0.81. This shows the data in a little bit of a different, from a different perspective. This is the difference between the kidney function predicted and the actual clearance of the medication. So if we start with eGFR, on average, eGFR is comes within about 33% of actual furosemide clearance averaged across the study. Right? So when we go to IGFR, a more direct measure of kidney function, that percent difference goes down. We're now getting closer to predicting actual furosemide clearance. When we use our summary secretion score, that gets even lower. So it was the best of the three measures, but that difference between directly measured GFR and our summary secretion score is relatively modest, only about a 6% improvement, and the confidence interval crossed to 0. Same thing qualitatively was seen with PENCICLOVIR. The best measure, the worst, from worst to best, eGFR was within about 30%, IGFR within about 24%. Our secretory clearance summary score, 18%. Again, not significant. So modest improvement, but not a huge difference. So to summarize these results, in stable persons with and without kidney disease, the kidney clearance of endogenous secretory solutes were highly correlated with the kidney elimination of avidly secreted drugs. But also in relatively healthy, well-appearing outpatients who volunteer for research studies, there was a pretty strong correlation between GFR and secretory clearance, roughly about 0.8 there between GFR and secretory clearance. So in these people who are relatively well, the two kidney functions are in fact reasonably correlated, and GFR does act as a reasonable proxy for kidney drug elimination. Again, marginal improvement of the secretory clearances for predicting the drug elimination. And when we combine the secretory clearance score with GFR, we're able to get even closer to the medication clearance. Again, these improvements were somewhat small, but suggestive that in the future, we might be onto something for improving kidney medication dosing. And so we were surprised by this. As I've talked about before, filtration and secretion are just so different in terms of their mechanistic underpinnings. So filtration is a passive filter. Things cross the filter if they're a certain size and charge, and don't if they're not. Secretion is this whole active process that requires cellular energy, it requires coordinated transport, polarity. People could differ and do differ by polymorphisms in these transporters, what kind of diseases they have. So we thought there'd be a lot more variability between GFR and secretory clearance in individuals. And there very well may be when we get into more different diseases, but at least in this group, it was not so much. It sort of suggests that blood flow and renal hemodynamics are really a primary driver of both processes. Limitations of this study: relatively small sample size, not really enough people with severe kidney dysfunction, and most importantly, as I've said before, relatively stable outpatients with stable kidney function, willing to participate in the study, a heterogeneous mix of kidney disease etiologies, and we only evaluated specific drugs. There's more drugs to evaluate. So we are very interested in how these kidney functions may or may not diverge in more significant disease settings, especially in diseases that pick on the tubules or pick on the glomeruli differently than the tubules, such that there would be more dissociation between the functions. And so we are planning to look at this in other populations, such as AKI or autosomal dominant polycystic kidney disease, where the functions are more likely to diverge. We'd like to look at more medications. We still think we can do better with our secretory clearances. We think we can get more precise measures, and maybe when we improve that, we can show more separation. And we have plans to look at in more detail at the genetics. Okay, so that's one of the studies that we've done with secretory clearance. I'm going to tell you about one other one and then wrap it up. Again, we've done lots, and always looking for ideas as to how to apply this for diseases. I think there's some great places to look at this. This was sort of a different direction. It was occurring in parallel with our drug pharmacokinetic study, which was to look at the clinical relevance of secretory clearance in a large population of patients with chronic kidney disease. So for this study, we evaluated people from a national cohort study of chronic kidney disease called the Chronic Renal Insufficiency Cohort Study. And we measured plasma and 24-hour urine concentrations of our secretory solutes in 3,406 people in this study at baseline. We used the plasma measurements and the 24-hour urine measurements to calculate the 24-hour kidney clearance of each solute at baseline. And we looked at the associations of those clearances with the subsequent progression of chronic kidney disease, defined by a 50% decline in GFR or the need for dialysis or transplantation. So one thing we saw is, despite the fact that GFR and secretory clearances are correlated, you can really see in a large cohort that there's quite a bit of variability among people or across people. This graph shows on the x-axis estimated GFR in the chronic renal insufficiency cohort study, and on the y-axis our summary secretion score. And the red circles, we just sort of did the equivalent of pediatric growth curves here. The red circles are people whose level of secretory clearance is in the middle 80 percentile for that degree of GFR. So a person here, say, right with the GFR of 50 and a secretory clearance score of also 50, that's sort of about average for that level of GFR. But we certainly see in the green circles, there are people who have very high secretory clearance relative to their level of GFR. And there's people who have very low secretory clearance relative to their level of GFR. We'd like to learn more about who those people are and why their secretory clearance is not typical. We've done some of that in the CRIC study. What's nice about the CRIC study is this large sample size, but with all these big studies, there's not a lot of granular detail. So we've found some things. People who have relatively low tubular secretory clearance relative to their GFR tend to have worse hyperparathyroidism, but not other mineral metabolism disorders that were measured in CRIC. They also have an unusual dyslipidemia with high levels of triglycerides and low levels of HDL on average, but not any differences in LDL. We couldn't really take it any further than that because those other more detailed measurements of lipid metabolism were not available in CRIC. They also tended to have high blood pressure relative to those with lower secretory clearance. So our main focus was how is measured tubular secretory clearance related to the subsequent progression of kidney disease over follow-up. So this shows the association of the clearances of the individual secretory solutes with progression of CKD, there were 1,206 progression events over a median of 6 years of follow-up. And again, progression defined by a 50% decline, dialysis or transplantation. And these hazard ratios represent the relative risk of progression after adjustment for GFR and albuminuria and other traditional risk factors. And so there's sort of modest associations of the individual secretory measures or secretory clearance markers with kidney disease progression, independent of GFR, albuminuria, and other risk factors. And the summary secretion score, again, showing a modest association here, though statistically significant, after accounting for traditional measures of kidney function. This slide shows the same associations, but this time with all-cause mortality. So there are 1,004 deaths over a median of 9.6 years of follow-up. And again, there were associations of the individual secretory clearances, mostly isovaleric-glycine, tigroglycine, to a lesser extent, cinnamol-glycine, and the summary secretion score, which is again the scaled average of these different secretory clearances, was also associated with all-cause mortality after adjustment for estimated GFR and albuminuria and other factors. So to summarize, in a national cohort study of adults with chronic kidney disease, we found associations of lower tubular secretory clearance with progression of chronic kidney disease and death, independent of these traditional kidney measures. And we observed relatively substantial variability between estimated GFR and secretory clearance in this large group. Of course, there's limitations in any big study like this. When we did the kidney clearances of the secretory solutes, it was based on a 24-hour urine collection and a single plasma measurement that was done in the morning. And as we've seen, these secretory solutes have variability in their plasma concentrations over the course of the day, which adds noise to the estimate of secretory clearance. In our drug studies, we were able to measure plasma levels of the secretory solutes at multiple time points during the urine collection and average them, which takes away that biologic variability. But we didn't have samples like that in the Crick study. Again, being a national cohort study of CKD, it was not focused on cause. Very few people had kidney biopsies. We don't know the underlying etiology of the kidney disease. And the Crick study excluded people with very severe kidney disease at baseline. So we don't know how these secretory clearances may operate in people with the most severe kidney dysfunction. Next steps, sort of similar to our drug studies, we really want to take a look at these measures in disease states in which we think glomerular and tubular functions are most likely to diverge. We want to explore further associations of secretory clearances with metabolic complications. We specifically saw differences in hyperparathyroidism and dyslipidemia. And again, we want to improve our estimates of secretory clearance to get more accurate and more precise. We have others, we've done many studies. Those are the two I really wanted to share with you. Those are the two I think that have sort of showed two different sides of where we're going with this. This work reflects the very, very hard work of a huge number of people, too many to even start to thank here, but here's a partial list of the many people who were involved in all of these studies and ongoing studies with relation to secretory clearance. I'm always interested in collaboration and applying these to different disease states. I think the more people who work on things, the better. So if there's ideas for how this could be used in particular disease settings or research questions, please let me know. Contact me anytime. I'm happy to talk and collaborate. And I'll stop here and take any questions. What about okay, I'm reading here on the Q&A. Okay, so what about the role of tubular reabsorption in clearance? Right, this is a great question. So the solutes that we evaluated are almost certainly, though not 100% certainly, but very, very likely to be secreted into the urine and not subsequently reabsorbed. So for secretory solutes, the ones that the kidney bothers to take from the blood and secrete all the way into the urine, those are not typically reabsorbed back into the blood. But reabsorption does, of course, play a role in overall kidney clearance for solutes that are filtered, because the more that it's reabsorbed, the less kidney clearance. That's a whole other aspect of tubular function that we really would like to evaluate in the future. And we're not, just didn't evaluate that in the studies here. Second question was, some of our diseases appear to affect tubules more than glomeruli, for example, IgG4. Can your secretory clearance predict the biopsy? So, right, we have ongoing studies to evaluate the association of secretory clearances with findings on kidney biopsy. To give you some preliminary results, yes, they are related to tubular interstitial fibrosis and atrophy in biopsy specimens. We are in the process of doing more analyses and getting more biopsy data for that. But this is a great example of where secretory clearance might be potentially a better measure of kidney function than GFR, which is less affected by those diseases. I got a question here. Do you see any advantage of using secretory measures in separating out pathology, disease activity, and lupus? So that's a great one. You know, lupus is obviously both glomerular and tubular, right? It's got pathologic components of both. And the temporal sequence is not 100% clear, right? We don't know sometimes which one might be coming first or second. So it would be very interesting to look longitudinally in people with lupus to see how those different kidney functions evolve over time and how that may correlate with lupus activity. We haven't done any work with that. It would be a great direction. The same thing is true with other. I mean, the thing is, you know, there are some kidney diseases that are very specific to one type or the other. Membranous is purely an attack on the glomerulus, whereas something like IgG4 is pretty much purely an attack on the tubules. But then we have all these other diseases that do, like lupus, where both are affected. Diabetes is another example. And I think there we have to do some careful temporal evaluations to see how these functions change over time with the disease processes. Let's see. It appears to be well correlated, but how correlated were OAT13 and OAT2 function across the two substrates? Right. So just based on the kidney clearance of these two drugs, one being an OAT13 substrate, the other being an OAT2 substrate, the kidney clearances of those two drugs were strongly correlated. Whether or not that might be true for other substrates and in different disease settings, I don't know. But in this study of 54 people, and with these two substrates, the correlation was something like 0.85 between the two different secretory drugs. What about drug effects on tubular transporters? It seems that large population studies don't have the granular information required to factor drug effects into the analysis. Absolutely. This is a whole exciting area, I think, in pharmacology, where you've got people on multiple drugs that impact the tubular transporters, and they may be competing for spots on those transporters. Although it does seem that there's a tremendous abundance of these tubular transporters, and they're relatively difficult to saturate. We've got some evidence for that in people with severe chronic kidney disease with extremely high levels of secretory solutes. There's still a lot of transporter. But certainly with these drugs that have different affinities for the transporters, there may be some competition, and some of them may be partially inhibiting or affecting the function of the transporters themselves. So that requires further exploration. Let's see. Interesting metabolites. What is the effect of the microbiome? Right. So, I had a study about slide about the microbiome, but I took it out. So let's just see here. So of our secretory solutes, the gut microbiome is involved in the production of many of them, although the final steps for the synthesis of all of these compounds does not occur in the microbiome, but occurs in the body, typically in the liver. our goal here is to find candidate solutes for which we can estimate their kidney clearances completely independently of their production. Because if the gut microbiome is influencing their production or other aspects of metabolism are influencing their production, if production has something to do with our measurements, then we'd be adding a huge amount of noise. In theory, when you're measuring the kidney clearance of a substance, it doesn't really matter to what extent it's produced. It should just be the amount that the kidneys are removing per unit time. Now that assumption is based on this idea that the plasma concentration over that period of collection is constant. Right now, in order for us to assure that, we have to get multiple measurements over that time period and average them, or focus only on the solutes that have very low variability in plasma. So absolutely, the microbiome has a role here.
Dr. Kestenbaum, it looks like there's one question that made it into the chat box from Dr.
Okay.
Mark Wenner. So it asks, can you get independent measure of renal blood flow? Examples are with MRI, MRA.
You can get estimates of renal blood flow with MRI or bold MRI measurements. You can do that, and so it's the jargon's a little bit tricky here about what actually we mean by actual renal blood flow, right? So just a quick step back. So we're actually exploring this, but we're going to instead of using MRI, we're doing this studies to estimate secretory clearance using radionuclide stuff using MAG3 renal scanning. So when we talk about renal blood flow, we talk about the amount of sort of uptake of a tracer by the kidneys. Often we're combining GFR and secretory clearance together, right? They're sort of mixed. And sometimes those studies include all the blood going into the kidneys, whether or not it's perfusing active living tubules or just going through the kidney and essentially being shunted, which is a big deal. So the studies like the MR studies can give you a sense of total blood flow to the kidneys, but don't necessarily provide a good breakdown of where that blood flow is going, if it's going to living tubules, whether those living tubules are doing the secretory clearance, and the distinction between what's filtered and what's secreted. I think I've made it to the, oh, wait a second, got more. Excellent, okay. So this one, regarding our SLE patients with minimal renal involvement, with preserved GFR and normal creatinine, are there any unique considerations we should keep in mind in terms of the renal handling of DMARDs compared with others with no, yeah. So could there be subtle tubular defects in SLE? Oh, there's really two questions there, right? One is that, is it possible that SLE is affecting the kidney tubules and we don't see any differences in GFR or albuminuria? The answer is it's possible. We might expect if there's enough damage to the tubules that we would start to see albuminuria, because the tubules play a huge role in the reabsorption of albumin that's filtered. And typically in diseases of the tubules, although you don't see nephrotic range proteinuria, you see proteinuria more so than micro. You tend to see proteinuria in the sort of 1 gram or less per day range. So I think if it got bad enough, you would see some albuminuria. Whether or not there's subtle changes in secretory clearance in people with normal GFR and lupus, that's something really interesting to look at. And then that would, of course, potentially alter the drug metabolism of the different medications used to treat. So all great ideas for research studies. So happy to take a look at these with people if we've got ideas for how to add these to existing studies or collect data prospectively.